Machine learning has changed the world forever, especially in the field of biotech research. We've seen tools like AlphaFold developed by Google DeepMind that have helped many researchers in their research and come up with radically new solutions to these biological problems. So what if we applied machine learning to one of the most successful forms of cancer therapy in the world? Well, this is what I did, applying my knowledge of machine learning to cancer immunotherapy. So what is immunotherapy? This is basically the science of engineering our immune system to fight several diseases, including cancer. There's a lot of forms of immunotherapy, including immune checkpoint inhibitors, antibody therapies, and what I focused on for this project, CAR T-cell therapy. T-cells normally work by interacting with receptors on cancer cells. This is called an interaction between a T-cell receptor and a cancer epitope. When this interaction is positive, the T-cell gets the signal to release cytotoxic molecules that kill the cancer cell. However, in actual cancer, this is not how it works, as these cancer cells produce several factors that make the T-cells unable to get that signal to kill the tumor. These include inhibitory cytokines, inhibitory ligands, and regulatory T-cells. So how can CAR T-cell help this? Well, CAR T-cell engineers an artificial receptor. This is made out of an antibody, which is then converted to a usable form called an scFv. These scFvs, combined with other domains, will be able to be transferred to the T-cell, and this creates the new T-cell receptor that can bind to specific cancer proteins that will allow the T-cell to get a greater signal and thus kill the tumor more effectively. You may be wondering, how can machine learning help us here? Well, we don't know what T-cells will actually interact with the cancer epitopes. We just know this by experimentation and knowledge that we've gained through different experiments. So if we knew how these T-cells interacted with these epitopes and could thoroughly predict this interaction, we may be able to speed up therapeutic discovery for these cancer therapeutics. So that's what I did. I built a machine learning model to predict the interaction between a T-cell receptor and a cancer epitope. So in this next part of the video, I'll be explaining the code and what I did to get here. The first step of building any machine learning model is gathering the data and engineering it to fit your use case. In my case, the project that I'm building has already been attempted by many people, and the database already exists for me to take from. This database is called the VDJ database, and it contains all the receptor data that I need. However, this is where I ran into my first problem. Scientists really don't like providing data on negative interactions, T-cells that don't interact with cancer cells. This is not very useful for main wet lab research, but for machine learning, this is super important, as you need to have those negative examples so these machine learning models know how to differentiate between them. To fix this problem, I had to find a machine learning model that could take only the positive interactions and be able to differentiate between those and the negative interactions. Luckily, I think I found just the thing. This is an isolation forest model. It basically works by splitting the data into several trees. And in these trees, it starts trying to separate this data that it gets from the data that it was trained on. So if you have an anomalous data, that means that it'll take lower amount of splits to separate it from the normal data. Whereas if you have normal data, then it will take more splits to separate it from other pieces of data like itself. So that means that the isolation forest model will determine if an object is an anomaly based on the number of splits it takes to separate it from other instances that it was trained on. So now that we have the model, it's time to start building. The first step is collecting the data. Now, engineering it and explaining all of that in this video would take too long, so if you want to check that out, please feel free to check out the article. But in short, basically I took it from the VDJ database, and then from here, I engineered it to tokenize into several numerical embeddings. This allowed the machine learning model to interpret these sequences as their complex structures, so need to have a numerical embeddings. So the machine learning model can actually understand what's going on and see all the features present in these proteins. So after the data was engineered, I had to move on to actually building the model. So here I have the code for the model, and I'm just going to do a quick walkthrough of how I built this model and how it works. So the first step is connecting to a GPU if it's available. And since I'm using Colab, I can use their free GPU storage where I can run this machine learning model to speed up the process of training and also processing the data. And after that, I need to read my file which has the data. And again, feel free to check the article in the description. So if you want to see how I engineered the data and how I created the data set, then feel free to check that out. And then after this, I had to read each column and then process the data so it can be passed into the model. After this, I can initialize the isolation forest model and fit it with the concatenated data that I have. And this creates the model that I have. And then from here, I can just create this data set for testing. And then after this, we could test the accuracy of the model. And as you can see, it's not very good. It's 67% about. And I'm planning to improve this by just using more data and including more columns possibly, including some other factors that may affect how a T cell interacts with an epitope. So if I do that, then I'll make sure to update this video and let you guys know. But other than that, thanks for watching this video. I hope you learned something new. And if you're interested more in the project, like I mentioned, there's an article in the description, which I think goes into more detail about how the model actually works and how I engineered the data. So if you're interested, feel free to check that out. But that's all for this time. Thank you.